Hello, we are from Group 4 Experiential Learning Phase 1 presentation based on the topic glucose sensors. Our team members are Rushil, Gopal, Sumit, and myself, Mallikarjun. So, now about the glucose sensors. Glucose sensors are mainly used to measure the glucose concentration in the blood, soft drink, juices, and many others. One of the most important part of the glucose sensor is that it helps in the managing of a diabetes mellitus. It is very important for managing of the blood glucose level. There are type 1 and type 2. These are the two types of diabetes which are most common in the normal people. Type 1 type of diabetes is usually diagnosed in children and young adults and accounts for 5% of all diagnosed cases of diabetes. Type 2 diabetes has been diagnosed in millions of Americans. According to diabetes report of 2012 issued by the National Center for Chronic Disease Prevention and Health Promotion, 18.9% of US adults over 65-year-old are diagnosed as diabetes in 2007 and 9. As the pictorial representation of this diabetes mellitus suggests, the first diagram is of the healthy pancreas which with a normal functioning which doesn't have any diabetes mellitus disease. In the type 1 case, pancreas fails to produce insulin. In the second case, insulin receptors are not proper functioning. So in the first case, there is no proper insulin is produced. Therefore, the lack of insulin is present in the type 1 case. But in the second case, insulin receptors are not working properly. So, which is this response for the diabetes mellitus type 2 disease. These are the major diseases which are must be diagnosed. Hence, glucose sensors are one of the most important machines or the devices that help in monitoring the glucose level in the blood. Majority of the blood glucose sensors are amperometric sensors. So the main function of amperometric glucose sensors are they measure the interstitial glucose values. And it means they measure the glucose level in the interstitial cells of our body. They does so by measuring the electrical current generated by the reaction of glucose either with the oxygen or with the immobilized redox mediator in the redox reaction or respiration in which food is broken down into energy, commonly said as the reducing property of a glucose is measured as a current. Thus, amperometric sensors work. As we can see in the diagram, the amperometric sensors work as follows. Working of amperometric glucose sensors. The majority of these sensors contains electrodes which measure the current generated between the reaction of the glucose, an enzyme and a mediator present in the cell which accepts a cofactor, which we will study in the biology of a cell. So the main reaction is the glucose oxidase catalysis. The glucose undergoes oxidation to produce glucolactone and hydrogen peroxide. So the sensors detect this current produced during these reactions. In order to act as a catalyst, glucose requires a redox factor called FAD, flavin adenine dinucleotide, and work as the initial electron acceptor and is reduced to FADH2. Based on this reaction and the current produced during this reaction, glucose sensors detect the blood glucose level in the blood.
Hello, I'm Sumit, and I'm going to be discussing about the types of glucose sensors. So the first one is the enzymatic glucose sensor, the first broad category, which again includes three other glucose sensor, which are first generation glucose sensors, second generation glucose sensor, and the last one is the third generation glucose sensors. And there's also another broad category of glucose sensor, which is the non-enzymatic based glucose sensors. So the first generation glucose glucose sensors actually use oxygen as a substrate, and on the detection of hydrogen peroxide, the first generation glucose sensors work. So measurements of hydrogen peroxide formation have the advantage of being simpler. So there's nothing very difficult about these methods to use. So it's just quite simple to use, and these are also smaller in size. So it's very convenient enough to handle these things, and but the problem with these first generation glucose sensors is that the operation of this biosensors actually requires relatively high potential of high selectivity. So to compensate the limitations of first generation glucose sensors, the second generation glucose sensor models were proposed. So the first generation glucose sensors was proposed by Flax from the United States of America, and the second generation glucose sensors were proposed by glucose sensors were proposed due to the limitations which were found in the first generation glucose biosensors, and this one actually use the second generation glucose sensor actually use the mediator substrates instead of hydrogen peroxide. These mediators help in the electron transfer between the electron transfer between the enzyme to the surface of the working electrode. So in the first generation glucose biosensors, the oxygen acted as the mediator. So in the second generation, mediators have a different work which actually play an important role in transferring of electrons from the enzyme to the working electrode. And in the third generation glucose biosensor, the mediator is replaced by another kind of material which is less toxic, and which performs direct electron transfers from using organic conducting materials based on charge transfer complexes. So these actually create some chemical complexes instead of those mediators in the second generation, or the as the oxygen in the first generation glucose biosensors. And yeah, that's it for the third generation glucose sensor. So the second thing is the non-enzymatic glucose sensor. Non-enzymatic glucose sensors use a completely different mechanism in detection of glucose level in the blood, and the active metal nanoparticle actually undergoes an oxidation step to form anhydrous oxide layer, and which actually mediates the oxidation of absorbed species, which in turn transfers electron from enzyme to the electrode. And this is the representation of first generation glucose biosensors. So we can see the conversion of the O2 actually liberate H2O2, which is hydrogen peroxide liberating oxygen, which in turn transfers electrons from the enzyme to the electrode. So this is a typical device used for the detection of blood sugar level. So which is an invasive method. So you have to just pinch through a needle or some instrument given in the instrument. So you just have to take a blood just drop of blood, and the device will detect the amount of glucose present in the blood. And the second one is the test strip, which actually you put the drop of blood, which in turn returns with the value of the amount of blood sugar level in the blood, glucose level in the blood. to the applications of glucose biosensors. So glucose biosensors actually provide quick responses to the glucose level, which actually tells us if the glucose level is high or low, which actually corresponds to the high, which actually corresponds to the high or low level of glucose content present in our blood. And the second one is it helps us to distinguish between hyperglycemia and hypoglycemia. Hyperglycemia is high blood glucose content, and hypoglycemia is low sugar content in the blood, low glucose content in the blood. They have and again they have been approved by the FDA, which is the Food and Drugs Administration of America, and commercialized such as Freestyle Libre, which is a company which manufactures all those glucose sensors. And there's another one called as Everseen CGM, which is an implant. So you just which is implanted under the skin and which automatically detects the amount of glucose present in the blood. And there's something called as Dexcom G6 CGM, which is again an implant specialized in the abdominal area. The implant will be placed in the abdominal area and which reads the glucose level. And the Guardian Connect system, and again once again it's implant which has to be put in the abdominal region. And these sensors are actually connected with smartphones, so that they can be controlled remotely. So these are some cutthroat technologies and recent advancement in the detection of glucose level in blood. So the typical way of detecting the blood sugar level was just the invasive method, in which you just pinch the just poke a hole in your finger and just draw some blood, drop of blood, and just measure the amount of glucose present in the blood, but which is not actually a very feasible way of detecting the blood sugar level. So there are many other ways, such as this. Such as the non-invasive method, in which you just have to the sensor actually detects the blood sugar level without actually causing any damage to the individual. So without drawing any blood from the patient. And there are also other gadgets invented to do the same work, which is to just detect the amount of blood sugar level. And these can be controlled through smartwatches and smartphones as well. And there's something called as oximeter, which detects the amount of oxygen content present in the blood. So the working principle of these oximeter and glucose sensor is almost the same. So these two are non-invasive method of optoelectronic biotransducers, which actually measures the oxygen level in case of oximeters, and glucose level in glucose sensors, glucose meters. And these are like the most recent advancement in this field to measure the glucose level in the blood. And in case of oximeter, is just to measure the oxygen level in the blood. The oximeter reads the refraction of the light and converts into pulse wave on the monitor. So this is how exactly the glucose level glucose meter also works. So they just use the optic basis rather than the actual content in the blood, like the one in invasive methods. So these were some of the applications of glucose biosensors. So thank you. I'll let you to carry on with the program of development of the glucose production center. Now I'm going to say about the program for development of Arduino circuit for low cash potential stat. Potential stat is a voltage source that is able to vary its output potential in response to changes in the resistance across a circuit. It is used in electrochemistry experiments, and these potential stats are able to supply more or less current so that the potential across an electrochemistry active cell remains constant. And also these potential stats control the potential between the working and reference electrode and measures the current between working and other counter electrodes. And a unified potential stat circuit for both O2 and H2O2 based glucose sensor was introduced, and its function was verified by circuit simulations and measurement result of fabricated chip. This is the potential stat circuit design. It consists of signal generator, feedback amplifier, control amplifier, current amplifier, and current ranges and signal converter. Signal generator determines the applied voltage resolution of the potential stat. The feedback amplifier is the most important part of the potential stat circuit. It measures the voltage between the working reference and the reference electrode, passing it to the signal converter, and it is sent to the computer. The control amplifier takes the voltage output of the signal generator and the feedback amplifier and outputs the voltage that will be passed between the working and counter electrodes. The current amplifier is used for measuring the current passing through the working electrode and the counter electrode. And the at last signal converter uses an analog to digital converter to convert the output of voltage feedback amplifier and current amplifier into digital signal that can be interpreted by computer connected to the potential stat. Electrochemical cell setup of a potential stat. This potential stat can be used to control 2, 3, or 4 electrode configurations. The 3 electrode configuration is by far the most common used setup, and also it consists of a working electrode, a counter electrode, and a reference electrode. Working electrode is the primary electrode in the system. It is where the applied voltage enters the system and where the most of the reaction and electron transfers take place. And the reference electrode is used in a three electrode system to maintain a stable potential against which the potential stat measures and controls the working and counter electrodes. The counter electrode is also known as an auxiliary electrode, where it completes the circuit of a 2 or 3 electrode system. I thought potential stat circuit, which is commonly used to set up the working electrode voltage and measure the current produced using the current sensor Arduino board. As the current passes through this shunt resistor, voltage is generated, and by using some analog values and some math, the current is determined, and then it is converted to a blood glucose value via the lookup table, which is stored in the Arduino memory. Arduino board connected to a battery and a glucose sensor. Next, Gopal is going to explain about the materials used in glucose sensing.
Let us discuss about the materials used in the glucose sensing. CMOS MEMS glucose sensors. These glucose sensors applies the working principle of first generation glucose sensor by detecting the concentration of the glucose oxidation. These are the two types of glucose sensing. First one, schematic of integrated glucose sensor. Second one is the cross section on the layer of the CMOS MEMS glucose sensor. Feature porous material. This is an experimental porous poly-L-lactic acid, which is known as PLLA, coating applied to the sensor tip of our investigational purposes. Advantages of glucose sensor. It is convenience, accuracy, easy medical decision, cost effective, is low, and other features. Next, disadvantages of glucose sensors. It is painful, non-compliance, not always accurate, might cause infection, result in skin changes. Thank you all.